Okay, it's really wonderful to be able to be here and give this talk, and also especially wonderful to be able to talk right after Carlo because he really introduced everything that, that I need to talk about. This needs to go back to the beginning. Okay, that's my talk. So I can get right into the data, and I have a lot of data, so I don't want to spend too much time introducing. The point that I just want to start by saying is I'm interested in talking today about metastatic lineages, how they can arise early, and how they can exhibit multiple genetic origins within primary tumors. And just to connect this with what Carlo was talking about, Carlo spent considerable time talking about diversity within tumors, and one of the approaches that colleagues and I decided to try to take to address this issue was to get around the issue of the fact that there is a lot of heterogeneity within tumors by actually looking at dispersed sites of tumors, that is, looking at metastatic sites and primary tumors simultaneously. And the advantage of that for looking at intratumoral heterogeneity is that since they are disseminated, you know that they went through some kind of bottleneck prior to their location in another location, and so you can actually assess the genetic divergence of those things particularly well. So I just want to very quickly introduce you to the data set. What we did was we whole exome sequenced 139 sites of metastases, plus normal and primary tumor tissue from autopsy patients. This is just a diagram of all of the different types of cancer that we assessed, the number of metastases assessed in each case, and then we also assessed normal tumor, normal tissue, and primary tumor, did all of the sequence calling. I got to brush over some of the methodologies here, but I'll just say that we had very good results. The tumor purity was high. 40% is actually high in the field, in case you want to know. Also, many of the reads were mapping to the genome and the exome. Just to keep moving, I just want to tell you what kinds of questions we're going to address using this data. So we're going to use phylogenetic analysis to address questions about the timing and relationships of metastases. And here are the questions I'm going to tell you answers to. Are there single or multiple genetic origins of metastases within the primary tumor? This is a very important question and addresses the linear versus parallel models of evolution that people have talked about for cancer over a number of years. How early do metastases genetically diverge from primary tumors? That's important, obviously, for therapy because you need to know whether you need to be thinking about metastatic lineages early in the treatment or only late in treatment of individual patients. What is the chronology of cancerous tissue origination? In other words, when does it actually happen? Did you have your cancer start diverging from your normal tissue 10 years ago, 5 years ago, 2 months ago, 20 years ago? And do driver mutations occur early or late in cancer? The current zeitgeist has been, as Carlo mentioned, to sequence a normal tissue and one sample from a primary tissue and say we're going to figure out the genetics of that. Unfortunately, that gives you no scope for understanding what happened historically along the evolution of the cancer. All right, so we conducted alignment of single nucleotide variants. They were concatenated. We made phylogenies robustly estimated by multiple inference procedures, and the inferences were consistent with loss of heterozygosity, for those of you who know this. And so these are two are the first phylogenies. I'm not going to go through them in detail. I really don't have time to do that, but you can see here there's a primary tumor in the lung. It was also, we had metastases in the lung, in the lymph node, the peritoneal soft tissue, the pancreas, the liver, the mediastinum, and that is actually the paraprostatic soft tissues. So a lot of different metastases. I'm going to talk about the structure of the tree shortly, so you don't need to spend much time on that. The main point here is just that where we saw loss of heterozygosity, it matched the phylogeny that we inferred. So we have independent data both confirming the phylogenies that we actually inferred. So I'm pretty secure that those phylogenies are correct. The model predictions are that there's a long-standing linear model of cancer progression that suggests that there would be a single metastasis-adapted descendant lineage in the phylogeny of tumors of each patient. That's the classic model of we have a primary tumor that originates, it develops in various ways and changes over time, and then near the end there's a terrible thing that happens, which is metastasis, and that is dependent upon some kind of mutations that occur for metastasis. A parallel model, rather, is consistent with multiple genetic origins of metastases. In other words, you can have one metastasis emerge early in cancer, another one emerge at a different time, and they aren't necessarily, in principle, as dependent upon particular mutations that lead to the metastases. So we can differentiate these by doing the phylogenetic analysis and address hypotheses about the singularity or multiplicity of origins of metastases within a primary tumor. And you can do this by looking at the number of tumors or metastatic sites obtained, and then look at how many different phylogenies are predicted to actually apply to that number of tumors. And you can, under the linear model, what you predict is that all of the metastases should be more closely related to each other than they are necessarily to the primary tumor. Under an idea of multiple origins of metastases within the primary will sometimes be nested within the metastases because the metastatic tissue may have departed from the primary tissue early, and the primary tissue that you sample may be more closely related to the metastases. So there's an expected fraction if this primary tumor just landed at random within the phylogeny. And you can calculate that by looking at the number of tumors obtained and using this simple formula for what the possible topologies are. And that allows us to assess this question. And the result of this assessment is that a linear model for cancer is not supported. So some genetic, epigenetic, or physiological disposition towards metastasis is, but this linear model is not. So there's a lot of numbers here. I'm just going to say it very quickly. Of 16 cancer phylogenies featuring a well-supported topological position of the primary tumor, 6 of them exhibited a most likely topology in which metastatic tumor lineages were not monophyletic. So they're all monophyletic in these 3 ones. This one, these is just an example of 4. In this one, we can see the primary is nested within the metastases and it's a well-supported phylogeny. So that implies that the linear model can't be applying as a general rule to cancer. Integration over Bayesian posteriors for all 32 phylogenies, that is where we take the uncertainty of the phylogenetic inference and just integrate over all of them, yields a very similar percentage, so 45%. You can see the confidence interval for that overlaps with the 6th where we had really well-supported phylogenies. This value is significantly higher than the random expectation of 21%. That's how much we expect if it were just randomly distributed. It's significantly higher, so it demonstrates that there's some heritable genetic, epigenetic, or lineage-specific events, it could be just physiological, that contribute a proclivity within lineages toward metastasis, but it does not agree with a lineage-specific effect that's so strong that it demands specific genetic changes for metastasis to occur. Similarly, if we look at when the genetic divergence of metastatic lineages occurred from primary tumors, we can see it can happen very early in tumor evolution, which also argues against very specific mutations occurring in order to engender metastasis. So in the cancer molecular evolutionary trees for 11 out of 40 subjects, the shared ancestral lineage of all tumors was shorter than the subsequent branch lengths leading to a metastatic tissue sampled at autopsy. So this is one example of that. I tried to just sample some of them to give you an idea proportionally of what kinds of trees we were getting. We inferred cancer chronograms then by applying a relaxed clock calibrated with the timings of diagnosis, biopsy, surgical resection, and autopsy, and parameterized by cell division times of primary tumor cells. And the result of that was that the first genetic divergence of metastasis usually predated diagnosis. That means the first genetic change that leads to the metastatic lineage happened before the person even was diagnosed for cancer, and that was true whether or not you looked at individuals who were diagnosed for metastatic cancer or just a primary tumor. Most of them were just diagnosed for primary tumor in this data set. Okay, so some examples of that. I'm just going to show you some phylogenies here. Metastatic lineage sampled sometimes arose subsequent to diagnosis. Here you can see the metastases arising subsequent diagnosis time. Sometimes they were arising prior to diagnosis and resection, such as this. Here you can see the resection happened less than a year ago, but there were many metastatic lineages inferred to have diverged quite a long time before that. The resection obviously did not capture those lineages. And metastatic lineages can also be 4 or even 7 years before. So this is an patient who was 4 years ago, had a primary resected. They came down with metastatic cancer, and you can see that they had lineages that had genetically diverged long before that resection. Same thing here. Here it's 7 years this first patient was, came back, and the metastatic lineages are there. All right, so conclusions from the phylogenetic analysis about the timing in relation to metastases. Divergence from lineages from the primary can occur early as well as late in cancer histories, and there are multiple origins of metastases within the primary tumor. We can also map known driver mutations to the tumorigenesis lineage prior to all tumor tissue or to the previous metastatic lineage. So this is very important. Where we map the mutations, you can find out, were they early mutations? Did they really originate cancer? And you can also look at this lineage where you might map them to see whether they are pre-metastatic mutations that led to metastasis. There was an enrichment of driver genes known to lead to cancer in this lineage compared to other lineages. There is not an enrichment of them here. So again, the evidence that we see does not point toward specific mutations leading to metastasis. It argues that it's more of a physiological process or an epigenetic process rather than a genetic one. So a couple just last points. Non-synonymous tumor type specific driver mutations, I also am not a big fan of that term like Carlo, but anyway, it's the one in the cancer field, are enriched in the early branch. So if you take known drivers, genes that are known to play roles in cancers of various types, and look at whether they're in that early lineage or late lineage, you have a very strong indication that more of those mutations occur in the early lineage than the late lineage, so that's what's really important in that case. And then non-synonymous mutations are enriched in those driver genes as well in the tumorigenesis branch. So if you look at non-synonymous changes, which presumably might actually change function and actually have a selective effect, versus synonymous changes, we get a very, very high enrichment of non-synonymous changes in known drivers compared to synonymous ones. And that's very significant by a Fisher's exact test compared to the other changes. And this is true when we remove the hypermutators or anything we do to this data, it still comes out significant. Last thing I just want to point out is that you can actually integrate over the uncertainty of that tree. Let me just back up for a second to explain how I'm doing this. You can take all of the trees, we've got 41 different phylogenetic trees here, and you can actually look at when these mutations are occurring at different branches, and then integrate over, and we don't know when it occurred along here, but since we have a 0 to 1 time scale here, we can say, oh, it happened in the first portion of the cancer or in the later portion of the cancer, and you can integrate over all that uncertainty to figure out when the order of cancer mutations occurred. And so what we get when we do that is this distribution. This is just assuming a beta distribution for when the probability of mutations occur. What I've done down here is line up the normal tissue. That is the first genetic difference of the cancer lineage from normal, which is different, which is not saying anything about whether it's cancer or not. It just says that lineage had its first genetic change that's different. So that's the beginning time on this axis. The end time is when they died. And what we did was look at when different genes were inferred to have mutated. So in these cases, this is the median of the branch of a particular phylogeny in which P53 was mutated, or KRAS was mutated, or PI3 kinase was mutated. And we mapped those to this 0 to 1 scale, and then inferred the distribution of when those mutations occur. And we can see that P53 and KRAS, two of the major drivers everyone talks about, are occurring very early in cancer, and this just reiterates that those are very important genes for us to understand better and work hard on. They're playing a very key role at the origin of cancer. PI3 kinase and KM2D, both also important genes, but they're occurring much later compared to those two on average, and ALK and KM2C are occurring even later. So what we really want to do is move from this data set, which was very broad cancer, it was a variety of different cancers, to look at specific cancer types, do the same kind of analysis, and it will provide really, really pointed information for specific cancers in where we should direct our therapeutic efforts in order to interrupt the evolution of cancer within patients. I just want to end, so hopefully there'll be time for a question or two, by thanking the people who have done this work. It's been a heroic effort after over 2 years. We're just now getting the paper out, hopefully, we're submitting it again. And Ximing Zhao led the whole thing and has done great work devoting her postdoc to this project. Stephen Gaffney did most of the evolutionary trees. Stephen Gaffney helped with the analysis of the sequence data, and Attila Iramoreno did the chronological inferences. So thanks.
Thanks. I've been told we have time for one very quick question. Alan.
Can I just ask, with the phylogenetic trees, am I right in thinking that there's a stepping stone model can be accommodated for that, so that you go from the primary tumor to one metastasis site to another, etc.?
Yeah.
So in other words, different metastatic sites could give rise to future metastatic sites.
So the tree does not tell us whether that could be. You could accommodate such a model to it, but we don't know that. In fact, one of the interesting things about this tree that I like to point out, which I didn't spend the time to point out, is, and I'll do this very quickly, I don't know what the primary tumor is on this tree. The primary tumor could be, I could be circling the genetics of the primary tumor here, right? So what we know about is just the genetic divergence, nothing about the physical departure of these metastases.
Thanks very much. I think we need to move on.